Gene therapy is the use of DNA to treat disease or disabilities.
I dreamed I was missing. You were so scared.
If gene therapy were to be used when a parent was expecting for a child, the doctor would test the baby for any disease or disabilities that it might have. If the baby came up positive for the disease or disabilities, the method would be used to correct the mutated or defective gene.
When I'm done here. So if you're asking me, I want you to.
The doctor or scientist will find the part of the DNA which needs to be fixed, then they will use an agent called a vector that carries the corrected DNA and injects into the cell. The vector will then find its way into the nucleus, replicate, and the cell's DNA is now corrected.
Don't resent me, and when you're feeling empty, keep me in your memory. Leave out all the rest. Leave out all the rest.
The scientist or doctor must first find the spot in the DNA in which the disorder is coded for.
My beating of shit, but I'm strong on the surface.
The scientist must then find a vector to carry the correctly coded DNA into the cell's nucleus. A virus is a commonly used vector.
So if you're asking me, I want you to know.
Once inside the cell, the vector will find its way into the DNA, replicate, and then the cell will carry the corrected DNA, and disability or disease will disappear. We follow the vector after it has penetrated the cell membrane. The cell membrane allows it to go through because the outside proteins on the vector are recognized by the cell membrane. It now goes through the nuclear membrane the same way. After it goes into the nucleus, it binds to nucleotide sequences of the DNA of the liver cell. After it binds, the new DNA corrects and it will replicate. The cell now has the corrected DNA and disease or disability are eradicated.
Some reasons to be missed. Don't resent me.
Can correct for defects and disabilities and can help treat and wipe out genetic disease.
Leave out all the rest. Forgetting all the hurt inside you.
Considered unethical by some, the safety of the procedure is unknown and potentially dangerous. The vector carrying the corrected DNA might not be completely rid of the disease and could actually infect the person. The dangers of gene therapy are very severe. If not conducted properly, the patient can receive the disease that is carried by the vector, which leads to very serious injury or death.